Hello, so in this video we're going to look at genetics and health, and the first little part is all about genetic crosses. So we've got some things that we're going to look at in this topic. We've got chromosomes up here, we've got a picture of somebody's thumb because there's a little question about how they've actually got a thumb which points forward or points backwards. We've got some more chromosomes, we've got P's because of this guy, Gregory Mendel. And we've also got this, which is the amniocentesis test, which is all part of genetic screening, which we'll look at later. Now, there's a few things that we need to link together from previous topics here. So the first thing is this idea about chromosomes. Now remember, chromosomes are found within the cell in the nucleus, and most of the time they're unraveled within the nucleus. But just before the cell divides, they'll condense, and we'll see something like this, which is a chromosome, and it contains these little banded areas which contain genes. The genes are short lengths of DNA which code for proteins. So in these videos we're going to be talking about genes, for example, blue eyes. So let's say, for example, the gene for blue eyes, it will produce a blue protein. And that's basically why you can have blue eyes or brown eyes. If you have the gene for brown eyes, you're going to produce a brown protein. Now, we also said that in each human cell, there are 46 chromosomes, or 23 pairs of chromosomes. So this shows a pair of chromosomes, and we say that those were homologous chromosomes. Now this pretty much means that these chromosomes carry the same gene in the same position. So if you look at all of these, you can sort of see how there's similarities between each gene. If you pick this one, number 7, you can see that there's a dark band up here and here, and then there's two sort of medium bands there and there. So just to look at those in a bit more detail, here's a pair of homologous chromosomes. Now remember, they only look like this just before the cell divides, but these are the things that we can see on them. So this whole structure, all of this is a chromosome. But just before the cell divides, the chromosome has to copy itself so that each cell ends up with the new DNA, and it becomes double-stranded, and each strand is known as a chromatid. So this entire chromosome is made up of 2 chromatids, and those are joined together by a central part known as the centromere. Then this little cell just shows us a typical animal cell with its chromosomes. Okay, so here we have the nucleus, and within here we have the chromosomes. And just to continue it, you can see here that we've got 6 chromosomes. So that means that we have got 3 pairs of chromosomes. Now, each chromosome here is made up of 2 chromatids. So altogether we've got 6 chromosomes, but 12 chromatids. Now the next part that we need to remember about is the adult life cycle. So each adult cell has 46 chromosomes per cell. Meiosis takes place in the testes and ovaries to produce gametes. The gametes will only have 23 chromosomes per cell, and they're known as haploid cells. Fertilization will take place where the sperm and the egg nuclei fuse together. This forms a zygote, which has 46 chromosomes, and that's the first cell of the new person. Mitosis then takes place, and they'll grow up into an embryo. Mitosis continually happens until they form a fetus, and then they'll form a child. And mitosis takes place for the purposes of growth and repair, and the child will grow up into an adult. And that was the human life cycle. Other bits we need to remember about is meiosis. So meiosis was cell division used for gamete production. It only takes place in the testes and ovaries, and the cells that are produced are haploid. Now that's going to be important in this example because if we have got a homologous pair of chromosomes, it basically means that only one chromosome from each homologous pair can go into each gamete. So that's going to be really important later on. Now what I mean by that is we start with cells which have 46 chromosomes and then we produce the haploid gametes which have 23 chromosomes each. And then this just shows us a little bit about meiosis. So we can see here that we start off with a diploid cell which has 46 chromosomes, but only one chromosome from each pair ends up in each cell. So we get daughter cells which have 23 chromosomes in them. Now remember, these daughter cells from meiosis were the gametes. Okay, so quite a lot of information we need to take forward, but let's have a look at this. And this is the whole concept of inheritance. So let's look at two people here. Now, the two people we'll look at will be John's mum and dad. You can see here that this is John's mother. Each of her cells contain 23 pairs of chromosomes, but on one pair of the chromosomes she has the genes for eye colour. She has 2 copies for the brown eye gene. John's dad has the same, only he has 2 copies for the blue eye gene. And there's their homologous chromosomes beside each other. So remember, you have 2 copies of the same chromosome, so they're homologous chromosomes. But when people produce gametes by meiosis, only one chromosome from each pair can go into the gamete. So John's mother has only the gene for brown eyes, so all her gametes will also contain this gene. And John's father only has the gene for blue eyes, so all of his gametes will also contain the blue eye gene. So they split up, and that's what you would get. So that means all of John's mother's eggs will contain a chromosome which contains a brown eye gene, and all of his dad's sperm will contain a chromosome which contains the blue eye gene. Now, fertilization takes place. Woo! And the sperm and the egg nuclei fuse during fertilization and form a zygote. So this is a little zygote. It's the first cell of the new person. It contains one gene for brown eyes and one gene for blue eyes. And it will grow by mitosis until a baby is born, and that's John. And then mitosis will continually take place until he's an adult. But that's not what we're concerned about here. We want to know what colour John's eyes are. So he's got one copy for the brown eye gene and one copy for the blue eye gene. So that means he should have one brown eye and one blue eye? No, definitely not. He gets one copy of the blue gene and one copy of the brown gene, but he has brown eyes. Now, in this example, that's because the brown gene is dominant and the blue gene is recessive. So in a way, the brown gene is stronger than the blue gene. So it doesn't matter that he's got one brown and one blue gene. The brown gene is stronger, so it is the characteristic that is shown. And we say it's dominant. And because the blue gene is weaker, we say it's recessive. Now, instead of calling these genes the whole time, we actually call them alleles. Okay, so there are different forms of the same gene, i.e. the gene for eye colour, because we can have a blue eye gene or a brown eye gene. Either way, they both control eye colour. So we call these alleles. So we've actually come across 3 important terms here: allele, dominant, and recessive. So an allele is different forms of the same gene, for example, blue eye gene or brown eye gene. Dominant is the stronger characteristic, and this is when individuals only need one copy of this allele for it to be shown. Recessive, on the other hand, is a weaker characteristic, and this is the important bit. If we want to see the recessive characteristic, the individual must have 2 copies of this allele, which is why in this example, John's dad has blue eyes, because he must have 2 copies of that recessive allele for it to be shown. Now, what happens next? Well, John grows up and mitosis takes place and he becomes an adult and he thinks, flip, I need to get myself a GF and I'm going to have a baby. So here's John with his beautiful brown eyes. Again, he's got 23 pairs of chromosomes, but on one of the pairs are the genes for eye colours. He's got one copy of the gene for brown eyes and one copy from the blue eyes. So he got the brown eye gene from his mum and the blue eye gene from his dad. And this is Jane. And each of her cells contain 23 chromosomes. Again, on one of the pair of genes are the genes for eye colours. This time, because she's got blue eyes, she has to have 2 copies of the recessive allele, or the blue allele. Now remember, when people produce gametes by meiosis, only one chromosome from each pair can go into the gamete. This time, because John has a brown-eyed gene and a blue-eyed gene, he can actually produce gametes which contain a brown gene or a blue gene. But Jane, because she's only got blue eyes and blue-eyed genes, all her eggs will contain the blue gene. So this is what we got. John's sperm can either contain the gene for brown eyes or the gene for blue eyes. Jane's eggs can only contain the blue-eyed gene. Now this means that John and Jane can have brown or blue-eyed children. And here's how. If this sperm fertilizes this egg, that child will have brown eyes. And that's because they have a copy of the brown-eyed gene, which is dominant. Or this child can have blue eyes because this sperm will fertilize this egg. So they end up with 2 copies of the blue-eyed gene. Now, in this case, there's some more terminology which we need to look at. Firstly, phenotype, which is their physical appearance. Now this is controlled by their genotype. If we focus on the first part, their physical appearance, in this example, it's basically what colour their eyes are. So John's phenotype is brown eyes, Jane's phenotype is blue eyes. The phenotype of this child will be brown eyes, and the phenotype of this child will be blue eyes. Now we said it's controlled by their genotype. The genotype is the combination of a person's alleles for a particular gene. Now it's usually represented by 2 letters, which we're going to look at slightly in a minute. Then there's also homozygous and heterozygous. So homozygous is when an individual has 2 alleles which are the same, and heterozygous is when an individual has 2 alleles which are different. So if we look at this example, John contains an allele for brown eyes and an allele for blue eyes. So we say that John would be heterozygous. Jane contains 2 blue-eye genes, so she would be homozygous. This child has 1 brown and 1 blue-eye gene, so they would be heterozygous. And this child will have 2 little blue ones, so it will be called homozygous. Now, to summarise that, homo means the same, hetero means different, and the zygous part comes from the zygote, which you'll remember was formed when the sperm and the egg fused together. So this would be a zygote, as would this. So a homozygous person means that they contain 2 alleles which are the same in their zygote, or in all their cells. Heterozygous means they contain 2 alleles which are different in their zygote. Now this just shows that idea again. So here we have a homologous pair of chromosomes, and on this they have different forms of the same gene which are present in both chromosomes. So in this example, there are 2 alleles which are the same. So in that case, this particular gene is homozygous, but in this case, they are heterozygous. Now, moving on a bit, if we look at this example of John and Jane, there's an easier way to work out what their children are going to have, and it's called a Punnett Square. So you set it out like this. We put their parents in, so the parents are John and Jane. We put their phenotype, which was their physical appearance. So John had brown eyes and Jane had blue eyes. We then put their genotype. Now, what we know about John was that John had brown eyes, but he had one brown gene, or one brown allele, plus one blue allele. Now we also know that the brown gene was dominant and the blue gene was recessive. So what we normally do is give each gene a letter, and you normally pick a capital letter for the dominant. So because he had one brown gene and one blue gene, we give him the genotype big B, little b. Jane had blue eyes, and she had 2 copies of the blue gene, so she's little b, little b. Now remember, only one chromosome from a homologous pair can go into a gamete. So the gametes that John can produce, which are going to be sperm, will contain a big B and a little b. So he can create 2 different types of sperm, one with the big B and one with the little b. Jane can only produce eggs with little b's. You take that information and we put it into a little Punnett square. So we put the female gametes here, which are Jane's. We put the male gametes here, and then we make a little grid and we combine them. So each of these boxes represents one child or offspring. So this one would be big B, little b, and try and put the dominant first. This one is going to be big B, little b. This one's going to be little b, and little b, little b. So in this cross, half of the children will be expected to have brown eyes, which are these 2, and half will be expected to have blue eyes, which are these 2. Now, try and use the same technique to show how 2 brown-eyed parents could have a blue-eyed child. Pause it here, see if you can work it out. So for this, their phenotype has to be brown because it said 2 brown-eyed parents. Their genotypes have to be big B, little b. Both of them do. And the reason why is because they want to have a blue-eyed child. So the blue-eyed child has to end up with 2 little b's. So each parent has to have a little b to be able to pass it on. The gametes that can be produced are big B and little b in both cases. So we add them into the Punnett square and we get this. Now, in this example, the ratio will be 3 brown to 1 blue. So our 3 brown is here, here, and here, and our blue is here. Now, let's look at some typical questions. Use the words homozygous, heterozygous, dominant and recessive to describe the following genotypes. So big A, because they're both the same, would be homozygous, and because they're 2 capitals, dominant. Big A, we're going to leave that for a second. We'll do little a. This one is homozygous, recessive. Now, the reason we're coming back to this one is because this one's heterozygous. But where people will make a mistake is they'll say it's heterozygous dominant. But it can't be that. We just say that it's heterozygous. Now the reason for that is hetero means that they're different. So we can't say it's heterozygous dominant or heterozygous recessive. It's just heterozygous. The next example is a black mouse mating with a brown mouse and all the offspring are black. Why are there no brown offspring? So use the symbols big B equals black and little b equals brown. What's the genotype of the black parent? Now, in this example, we're saying that when you take a black and a brown mouse, all the offspring are black. So therefore, black is dominant, which is why it's the big B. Using the symbols big B equals black and little b equals brown, what is the genotype of the black parent? Well, the black parent must be big B, big B. And that's because if it was big B, little b, you would have some offspring which would be brown. Now here's why. If the parent was big B, you'd be crossing, well, let's do it properly. Black crossed with brown. The brown must be two little b's. You work out the gametes. So because there's two big B's here, the only gametes they can produce are big B's. This one can only produce gametes with little b's. We put them into a Punnett square. And we get this. All the offspring end up with big B, little b. So they're all black. But the second part I said was if it was big B, little b, some offspring would be brown. So let's test that out. If it was black, cross with brown. And this time it was big B, little b. Cross with that. The gametes this one could produce are big B, little b. But this one can only make little b's. We put them in a Punnett square. That one's black. That one's black. Oh, but that one's brown and that one's brown. So 50% are brown and 50% are black. So in this example, the genotype of the black parent must be big B, big B. And the genotype of the offspring, all offspring are big B, little b. They're all black, so they all have to have one big B. If two of the black offspring from question 2 mate with each other, so let's set it out, black cross with black, and the ones we're looking at are big B, little b. What kind of offspring would you expect and in what proportions? Use a Punnett square to illustrate your answer. So this was our phenotype. This was our genotype. The gametes that we can get are this, cross with this. We put them in a Punnett square. We get big B, big B. Big B, little b. Big B, little b. Little b, little b. Now, in this case, there are 3 black to 1 brown. So our black ones are this, this, and this. All the ones with the big B. And the brown one is this one. The next part, a plant has 2 varieties, one with red petals and one with white petals. When these 2 varieties are cross-pollinated, all the offspring have red petals. Which allele is dominant? If they're cross-pollinated and all the offspring have red petals, that means that red must be dominant. So in this case, the alleles, or the letters you would use, a big R would be red and a little r would be white. Now, usually if you're talking about different forms of the same gene, so i.e. alleles, you use an uppercase or capital for the dominant and a lowercase for the recessive. Then pause it here and see if you can complete these crosses and just check your answers off. And set it out like this. So they're phenotype, genotype, then the gametes, and then work out the Punnett square. And then finally, give the offspring ratio of tall to short. So if we've got here heterozygous crossed with heterozygous, it's going to have to be big T little t crossed with big T little t. So their phenotype is going to be tall crossed with tall. Big T little t, big T little t. Those are the gametes. And the Punnett square. There's a tall one. There's a tall one. There's a tall one. There's a small one. So the ratio is 3 tall to 1 small. The answer for this one is all tall. And the answer for this one is again all tall. So just check that you've got those. If you haven't, you must have made a mistake somewhere. So work out from the start what their genotypes would be. So that's going to be big T, big T. That's going to be little t, little t. And then work through it until you work out that they're all tall. Now just remember, even though the answers are the same for this one, you can have different genotypes if you're tall. So if a plant is tall, it can either be big T, or big T, little t. And that's the end of this first section. Thank you. Goodbye.